Thank you again for attending our first Knauss Fellow Lunch and Learn, and my name is Jory Barley as a reminder, and I'm a current Knauss Fellow with the NOAA Center for Satellite Applications and Research. It's my pleasure to introduce our next speaker, Elise Keister, Dr. Elise Keister, excuse me. She is a current Knauss Fellow working as the main point of contact for the Ocean Acidification Program's international activities and serving as a secretariat for the Global Ocean Acidification Observing Network. She also provides support for early career professionals in working in ocean acidification via peer-to-peer and the International Carbon Ocean Network for Early Careers. Elise has recently completed her Ph.D. in biology at the University of Alabama, studying coral-dinoflagellate communities in the Florida Keys and Palau. She was supported by the Office National Marine Sanctuaries as a Dr. Nancy Foster Scholar. And with that, I will turn it over to Elise.
Thank you so much, Jory. Just give me one moment. Let me share my screen. Okay. As Jory said, hi everybody, my name is Elise, so happy to be with y'all today. So I just finished up my PhD at the University of Alabama at Birmingham, advised by Dr. Dusty Kemp. And I'm currently, in the Ocean Acidification Program of NOAA. So we'll just go ahead and dive right in. So today I'm going to be talking about my research, one of my dissertation chapters that was actually centered in Florida, and we're going to centered in Florida around Florida framework corals and their legacy effects. So just to kind of set the stage, coral reef ecosystems are incredibly diverse ecosystems, and 25% of all marine life rely on coral reefs at some point during their life stage or in their life histories. And it's an important source of protein for coastal communities. Coral reefs provide coastal damage mitigation, and then are also a really important economic source via tourism, fisheries, lots of things. So these coral reef ecosystems are incredibly important to coastal communities. And the coral holobiont actually forms the foundation of those coral reef ecosystems. Now, when I say coral holobiont, we're talking about the entire ecosystem of the coral, which is composed of the coral host, the coral animal host, as well as the microbiome. So similar to all those microbes that actually Katie was just talking about, as well as zooxanthellae. And those zooxanthellae are actually a type of symbiotic dinoflagellate. And from here on out, I'm going to be referring to those zooxanthellae as the symbiotic dinoflagellates. Now, these symbiotic dinoflagellates are really important for corals because they provide the corals with a majority of the energy that they need. So these dinoflagellates live within the coral tissue, providing carbon via photosynthesis, and in turn, the coral is actually providing nutrients to those symbiotic dinoflagellates. And this symbiosis between these dinoflagellates and coral is really what has allowed tropical corals to thrive in these really nutrient-poor waters for millions of years. And just to get an idea, so I know this is a little overwhelming, this phylogenetic tree, but let me just orient you a little bit. So we have here, this is the family of Symbiodiniaceae. So these are all the symbiotic dinoflagellates that exist. And they actually associate with lots and lots of different marine organisms, marine invertebrates specifically, such as jellyfish, anemones, different giant clams. And today I'm going to be talking about 4 genera that associate with corals. The first is Cladocopium, the second is Breviolum, the third is Duristinium, and then finally Symbiodinium. Now, coral reef ecosystems are incredibly colorful. I talked a little bit about the diversity. A lot of marine life relies on them. But unfortunately, we start to see a breakdown in ecosystem services as well as just ecosystem function due to stress. And unfortunately for coral reef ecosystems, that's a result of elevated temperature. And what we're seeing over time, if we move along the x-axis, is that the heat content is actually increasing as time goes on. So this heat content anomaly, this is measured in joules, and we're seeing this increase really starting mostly in the 1990s, and then it's increasing at this really steady rate. So this is kind of bad news for corals. And what we're seeing is that this temperature stress is creating a lot of issues. And this is, we're actually in the middle of another mass bleaching event. There's bleaching happening at the Great Barrier Reef. There was bleaching that happened in the Florida Keys. And I know that's okay. So, and then we have coral bleaching. So I mentioned a little bit of bleaching. So here we have on the left, this is a healthy coral reef pre-stress. So it is nice and colorful. But then when we have a stress come, like a thermal stress event happen, the coral actually becomes this stark white color. And what is actually happening is we're going from the coral living in healthy symbiosis with those symbiotic dinoflagellates that I mentioned, and providing a bunch of pigment for the color, pigment to the coral, and then providing all this carbon to the coral. But what happens during thermal stress is those algal cells actually get expelled from the coral, and it's a breakdown of that symbiosis that we're really witnessing. So we're not seeing those symbiotic dinoflagellates within the coral tissue. We're looking actually straight through the coral tissue at the coral skeleton. And at this point, the coral is still alive, but what we're seeing is it really being like physiologically stressed, both the algae as well as the coral. And unfortunately, we're seeing that coral bleaching is becoming more widespread. So the number of locations experiencing coral bleaching is increasing over time, with the first real big event that happened with the 1998 El Nino event. And then we're seeing the number of unbleached locations here in blue is dropping as time goes on, but the cumulative number of events is actually increasing over time. So this is a lot to take in, and it definitely seems very doom and gloom, and I want to provide a little bit of hope. So my dissertation really focused on how corals can tolerate thermal stress. Or you can maybe think of it how corals can kind of combat thermal stress. And so there are several resilience attributes that corals have. So one of them is the ability to associate with the thermally tolerant symbiotic dinoflagellates. Another is for the coral to increase heterotrophic feeding. So if the coral is able to feed on plankton, it's not going to need that energy provided from their symbiotic dinoflagellates as much. Another resilience attribute is being able to maintain high energy reserves, and then being able to use those high energy reserves and break down those molecules during a thermal stress event. And what's interesting is we see a trend kind of worldwide where we see nearshore reefs experience a warmer temperature on a regular basis compared to offshore reefs, which experience cooler temperatures. And this is just a generalization, but what we're seeing is that the nearshore corals, because of this regularly experiencing elevated temperatures, they're a little more resilient against warm temperature events. They're less likely to bleach, or if they bleach, it's less severe. Mortality is a lot less. And because of that, my dissertation really focused on what are these nearshore communities doing that's so different from the offshore communities. And so to just kind of dive in, this work was all centered in the Florida Keys. And the main question I wanted to answer was, do tissue biomass and energy reserves, are these actually contributing to the nearshore coral resilience? So to answer this question, let's just zoom in a little bit to the Florida Keys. So my first site was an offshore site known as Little Grecian that has a pretty narrow temperature range of about 18 to 30 degrees Celsius. This is buffered by the Gulf Stream, which is what allows this temperature range to be a little smaller. And the coral cover here is about 3%. And here is an image. You can see the water is nice and clear. Now, when we go to the lower Keys, our nearshore site, which is known as Jap Reef, the temperature range is much wider. So it's 16 to 33 degrees Celsius. So we don't have that buffering that's happening, and it gets much colder in the wintertime and much warmer in the summertime compared to the offshore site. But the coral cover is considerably higher. And here is a photo. This is actually, believe it or not, a pretty clear day. That's a pretty turbid reef, and this is likely also contributing to the resilience. So I mentioned that the nearshore site experiences this wider temperature range, and that's because the corals in the Florida Keys exhibit seasonality. So to answer this question, the questions I posed before, we also wanted to get an idea of what's happening over time. How are these variables changing from winter to summer? So I sampled in December 2019, summer, October 2020, October 2021, and then in March 2022. And this was really to get at this idea of seasonality. And unfortunately, there was a visible bleaching event that happened at the nearshore site in October 2020. So this provided an opportunity to get a better understanding of how these communities are really responding, where they are before a thermal stress event, what they're doing during, and then following. So we're able to potentially track recovery. And just to provide a little bit more information, here we have time on the x-axis and we have temperature, and this is for our offshore site. The little asterisks, these denote bleaching events. So there were two back-to-back bleaching events that happened in 2014 and 2015, where we have these dotted lines. This is where I actually had to use satellite data to fill in this temperature data. We got the temperature data from FWC down in Florida. And here on the secondary y-axis, we see something called a degree heating week. And essentially what that means is, the higher the degree heating week, the more stress, essentially, the coral is experiencing, the more thermal stress. And so what we're seeing is, going back to 2014, 2015, these are the last documented times that these corals were thermally stressed and bleached specifically. So we didn't see anything happen from 2019 until 2020. Now, in contrast, our nearshore site, we have more gaps here. So we have that satellite image, or the satellite data, luckily filling it in. But these corals also bleached in 2014 and 2015. And then I added an asterisk here for that visible bleaching that we saw in 2019. And here, the temperature range is more than what we see offshore. And notice the degree heating week, it's a totally different scale. So it goes all the way up to 16. So in general, these corals are experiencing much more thermal stress. And then just to get an idea, let's put these two together, and you can have an idea that the nearshore corals are experiencing warmer temperatures more regularly, colder temperatures more regularly as well in the wintertime. So these are very different ecosystems, and these abiotic factors and temperature is considerably different. So I looked at two of Florida's framework corals, both Orbicilla species, so Orbicilla annularis, as well as Orbicilla babulata. So I'm going to go ahead. Oh, yes. Okay, perfect. So the way I went about answering these questions is I looked at tissue biomass. So this is a measure of total organic content within the coral. I also, within that, we have our energy reserves, which include total lipid. And then within our total lipids, we have different lipid classes. There's also soluble protein, and then finally carbohydrates. So our total lipids, soluble protein, carbohydrates all make up our total energy reserves. Then I also looked at the symbiont cell density. So these are the number of cells of those symbiotic dinoflagellates that I've been speaking about, as well as the chlorophyll A content. And finally, we also looked at those symbiotic dinoflagellates. We looked at their community composition at the general level. And what's cool about, or more unique about Orbicella, is that their symbiotic dinoflagellate communities alter. So pre-stress, or when there's not any stress event happening, these are going to be mostly dominated by Symbiodinium species, Breviolum species, as well as Cladocopium species, with a distribution really depending on where you're sampling, if you're sampling a shaded versus a sunny area of the coral. But when there's a stress event and bleaching happens, we start to see shifts in these communities. So we still have Symbiodinium species, and that's present throughout the colony, but then we start seeing Durasdinium species pop up. And then during the recovery stage, we're still seeing Duristinium species and Symbiodinium species. And then we're kind of reverting back to this more stable association that the orbicellas have with Breviolum. So this is a trend that we see in orbicellas. So not only can we look at whether bleaching happened or not, we can look at their community composition and get an idea of where those corals are physiologically. And the Symbiodiniaceae, so this is that family name of the symbiotic dinoflagellates, this physiology really is playing a part, as well as the host physiology. They're interacting with each other. And ultimately, when we have a higher temperature event, this is going to, how these two physiologies play or interact with one another, this is going to be really dictating the bleaching susceptibility of the coral. So I'm going to dive right into Orbicella annularis. So this was looking at our symbiont community composition, and each column is essentially one individual. And what we're seeing here in December 2019 in Orbicella annularis from our nearshore colonies is that these corals are mostly dominated by Breviolum species. And then as time goes on, so October 2020, this is when our thermal stress event happened. So we're starting to get more of these mixed communities, just like how I showed when we see during the stress. We're seeing more of these mixed communities, less dominance by Breviolum, and this is something that we're seeing. So this is kind of confirming this that these corals are physiologically stressed. And these mixed communities are being maintained a year later in October 2021. And then we have this reversion back to these more stable symbioses in March 2022. Now, in contrast, at our offshore location, again, the columns show one individual. We're starting off the bat having more of these mixed communities that are dominated by Duristinium, really indicating that some type of thermal stress event happened, or some type of stress for the corals. And then these mixed communities are persisting even a year later. And it's not until October 2021 that we're seeing these more stable symbioses. And then that's maintained through March 2022. And when we look at the cell densities, again, these are for the symbiotic dinoflagellates, we're seeing this drastic drop in the near shore from December 2019 to October 2020. This is when our bleaching happened. So this is what we're expecting. And then we're not really seeing big changes over time. There is a slow increase for near shore over the course of our time points. And then we have a decline in general from offshore from our first time point to our last time point. And we're seeing at both nearshore and offshore for our chlorophyll A, it's slowly increasing from December 2019 until October 2021. Okay. And so when we look at the ash-free dry weight, or the total tissue biomass, we're actually not seeing any significant changes over time at either site, which is interesting. So this is telling us that overall, our total organic content isn't changing. But when we dive in a little bit more and look at our total lipid content, which is measured in kilojoules per gram of ash-free dry weight, we're starting to see some differences. So nearshore, we're seeing that in October 2020, we're seeing a significant decline, likely due to this thermal stress event that we witnessed, and then a slow build up over time in October 2021, and then right about at the same level in March 2022. And we're seeing no significant differences offshore. So this is really interesting, seeing this, being able to pick this up in the lipid, even though we're not picking it up in the tissue biomass. And then when we look at the wax esters, which are a really important storage lipid for corals specifically, and this actually makes up a large percentage of the lipid classes of the lipid for corals, we're seeing a sharp decline near shore, again, during that stress event that we witnessed. And then a year later, it's back up to where it was pre-stress levels. But we're also seeing a bit of a decline here offshore as well during this October 2020 time point, which is very interesting. And when we look at sterols, which are an important structural lipid in corals and are often correlated with higher heterotrophy, so when there's more sterols present, it tends to be that the corals are feeding more on plankton. We're seeing an increase, at least in the sample distribution of sterols in October 2020 at the nearshore site, and then a little bit of a slight decline offshore. Okay, so here we are with our little summary slide. So what we're seeing with the symbiont communities is that near shore, they start off stable, become mixed, and then we have that reversion back to the stable symbiosis, likely indicating these corals are able and still recovering. But what we're seeing offshore is that they're starting off mixed and then they become stable. So that's suggesting there was some type of stress event that happened before. Ash-free dry weight is equivalent for both. Across all time points. The total lipid declines near shore in October 2020, and we're not seeing any differences offshore. The wax esters are declining both near shore and offshore during October 2020. So there's something that's physiologically stressing corals at both sites. And the sterols are equivalent near shore, and offshore, they're declining in October 2020. And then finally, our phospholipids are equivalent kind of across the board. And then our protein content is equivalent near shore and increasing offshore in October 2020. Now, to switch gears, let's look at Orbicella fabulata. So these graphs are all the same. So there's similar setup as before, with one column being one individual. So near shore, what we're seeing is that we're seeing more of these stable symbioses dominated by Breviolum in December 2019. And we're seeing more of these mixed communities, again, during this visible bleaching event. And then 1 year later, they're able to switch back to their stable symbioses. And then in March 2022, we're seeing a little bit more mixed communities. And offshore, we're seeing something really different happen, where even from Orbicella annularis, where we're starting off and maintaining these stable symbioses over time throughout all 4 of the time points, which is likely indicating these corals were not physiologically stressed during any of these time points or in kind of recent memory. And what we are seeing is that we're picking up that the cell densities are declining from December 2019 to October 2020, which is what we expect because those cells are being expelled during that visible bleaching event. And we're seeing kind of this slow recovery over time. And in contrast, offshore, we're starting at a lower cell density and then increasing. And then finally, with our chlorophyll A content, we're seeing it starting off low near shore, and then by the time we get to October 2020, the chlorophyll A content is up. And then in general, offshore, we're just seeing this decline in October 2020, but this also follows those seasonal patterns that we see where chlorophyll A gets a little bit lower in the summertime compared to the wintertime. And when we look at the ash-free dry weight here for Orbicella fabulata, we didn't see any significant differences with annularis, but here we're seeing a decline in total tissue biomass in October 2020. So this is when that visible bleaching event, again, this is really hitting that. We saw them stressed, we saw their community shift, and their actual biomass is declining as a result of that stress. And when we look at the total lipid content, we're also seeing the same thing, where we're seeing a decline in October 2020 that's maintained even a year later. But by the time we get to March 2022, that is back up. And then we're seeing no significant differences offshore. And our wax esters are giving us an even clearer picture of what's happening with the corals. So we're seeing a sharp decline in October 2020 in the nearshore corals. And then for the offshore corals, the wax esters are starting off lower. And then that's maintained through October 2020. And then that goes back up by the time we get to October 2021. And these wax esters, again, are then an important storage lipid and make up the largest percentage of lipids in corals. And we look at our sterols, really the thing that jumps out the most is that this really high variability in sterol content in October 2020. Which could be meaning that these corals are tapping into and like feeding more heterotrophically on plankton in particular, or organic matter in the water column. So what we're seeing with our symbiont communities is that they're going from stable to mixed, but then they're able to revert back. And offshore, they're just being maintained. And we're seeing a decline in ash-free dry weight in the nearshore site, but no differences in the nearshore site, no differences in the offshore site. Our lipid content is declining nearshore, but nothing's happening offshore. And our wax esters, in contrast, are declining at both sites in October 2021, and they're lower in December 2019 for the offshore site. And our sterols are increasing in October 2020, and our phospholipids are declining in October 2021. And then we're seeing really different things happening with our protein content. Overall, what we're seeing is that this offshore site, which doesn't experience as extreme temperatures as our nearshore site, looking at Orbicilla annularis and Orbicilla fabulata, we're seeing wax ester declines, particularly during that October 2020 time point. Offshore, we're seeing a persistence of mixed communities. So these are going to be communities that are more dominated by Duristinium and Symbiodinium. And we're also seeing this delayed recovery, likely from a previous stress event that happened before sampling began. And in contrast, what we're seeing at the near shore is that we're seeing these shortened periods of mixed communities. So the communities tend to go from stable to mixed back to stable, and we were able to capture that over the course of this study over those 4 years. And we're seeing this faster recovery as a result. And we're also seeing this increase in sterols. So what this is kind of telling us, or what the data are showing, is that there's this legacy effect that's, we think, is playing a part. So because those nearshore corals are able to be more resilient and bounce back, recover faster from these thermal stress events, they're better prepared and better able to withstand future stress events compared to their offshore counterparts. And this could be contributing to differences in coral cover across like these nearshore and offshore sites. There was a mass bleaching event that I mentioned that happened in the Florida Keys. I'm sure many of you on this call witnessed it firsthand. I wasn't actually there in Florida, but it was horrifying seeing all the corals bleached, and the temperatures got up to 37.2 degrees Celsius. And it was really the warmest it ever been. And I'm pretty sure, the whole degree heating weeks, a lot of that had to be revamped because it was something that had never been seen. The temperature, the duration of the temperature, all very scary. So I think research focused on corals that are already able to kind of withstand higher temperatures, those are going to be, it's going to provide a lot of insight to what the future of coral reefs are going to look like. And with that, I want to thank all my funding sources, including UAB and then the National Marine Sanctuaries in the Florida Keys for all the permits, and then also the Office of National Marine Sanctuaries for supporting me as a Dr. Nancy Foster Scholarship. Here are all my committee members. A big thank you to them. And yes, with that, I will take any questions.